When a bacterial parent cell goes into binary fission and enters the elongation phase, it's going to have to synthesize both new cell membrane and new cell wall as it gets longer and longer. Now, synthesizing new membrane isn't all that difficult. If you recall, the individual phospholipids that are in the membrane are not bound to each other. The membrane is very fluid and loose. So new phospholipid molecules can simply be added to the existing membrane. In comparison, the cell wall is actually a very rigid structure, and the individual subunits in the cell wall are attached to each other. So in order to make new cell wall, the parent cell will have to cut the existing cell wall and then add new subunits of cell wall into the broken place before finally sealing up the gaps between the new and the old. It doesn't matter what shape of bacterial or archaeal cell we're talking about, that new cell wall has to be inserted using the same process. If you look at the process in a coccus-shaped cell, you can see this process under the microscope quite easily. The new peptidoglycan is going to be synthesized bidirectionally from the center outward. In comparison, when you watch a rod-shaped cell undergoing elongation during binary fission, new peptidoglycan will be added at several points along the length of that cell. Recall what peptidoglycan is made out of. There are two very special types of carbohydrate in that molecule. One is called NAG and one is called NAM. And long chains of NAG and NAM are built, alternating one after the other. And then those long chains are cross-linked to each other using short branches of amino acids and peptide bonds holding those branches together. So when a parent cell is going through elongation and needs to make new cell wall, it has to cut the old cell wall and then deliver new NAG and NAM molecules into the break. Those NAG and NAM molecules are being made inside that parent cell. So that means the parent cell has to get them across the membrane through that little bit of space called the periplasmic space, and then into the broken area of the cell wall. It's a several step process. Now, if you look on the right-hand side of this slide, up at the top, you can see an artist's drawing of a coccus-shaped parent cell undergoing elongation. And what's been drawn here in this brown beige color is the cell wall. So you can see that the parent cell has cut into the cell wall. It's severed the old cell wall, and the green membrane can be seen underneath. And the membrane is expanding and expanding as this cell gets longer and longer. You can see that exact process going on in this image at the bottom that was taken with a scanning electron microscope. It's pretty amazing, isn't it? You can see that the parent cell, this would have been the original parent cell, and this would have been the copy. And this area in here you can see is dividing. And you actually have these two new daughter cells also going through binary fission, and the septum has started to form in both of them. It really is an amazing process, and it involves several different protein molecules and a molecule called a carrier molecule to help get the new cell wall built. So let's take a look at these key molecules that allow for new cell wall synthesis to occur. What you're looking at over here is a very complicated looking diagram. It's actually just a piece of a bacterial cell. So we're looking at a little piece that cut out of a bacterial cell in the membrane. This is the inside of the cell. This is the cytoplasm. Then we have this little space here outside of the cell, and then we hit the cell wall. So the artist has drawn two strands of peptidoglycan, and you'll see that there's a capital G and a capital M, and then a capital G and then a capital M. This represents NAG and NAM. And remember, they alternate one after the other in a long chain. And then there's a second chain down here. Off of each chain, off of the NAMs, there are these little short branches of amino acids. Those are shown in red. And then the two branches are joined with what's called a peptide bond. So now we have these cross links between the long chains of NAG and NAM, and that's really what gives peptidoglycan its strength. So in the picture, what's happening is some new NAG and NAM, you can see the M and the G here, some new NAG and NAM has been built. The old peptidoglycan has been cut. We need to get the new NAG and NAM into this break here. So the first molecule is referred to as an autolysin. If you break that word down, auto-lysin. Remember, lyse means to break or to burst, and auto means self. So an autolysin is a type of an enzyme that can break old NAG and NAM strands. And it's the autolysin or the autolysin that does that breakage to allow for, again, that cell wall to split so the cell can elongate. The next molecule involved is called bactoprenol. And in the drawing, the bactoprenol is shown in orange right here. Now, bactoprenol is really interesting. Bactoprenol is capable of carrying the new NAG and NAM across that cell membrane and up into the periplasmic space. That tells you a little bit about bactoprenol, doesn't it? Remember, we said the kinds of things that can get across cell membranes have to have certain chemical characteristics. And one of them is it has to be non-polar. The hardest part of the membrane to get across is the tail region. And remember, the tails of phospholipids, shown in gold, are the non-polar parts. Remember, they're held together with van der Waal interactions because they're non-polar. So this molecule has to have some non-polar chemistry in order to get across that tail region. It's going to deliver the NAG and the NAM up here into the periplasmic space and insert it right into the place where it's broken. So we're building new cell wall. Now there's a type of an enzyme that does that work called a transglycosylase. Transglycosylases are enzymes that insert new NAG and NAM units into the old cell wall and allow it to grow. Now there's one more important process that has to happen. When we insert the new NAG and NAM in there, we have to build new cross bridges. And that's a process that gets a name. It's called transpeptidation. So in this drawing, what they're showing us is the nag and nam strands are running this way. Here's another one over here running this way. And again, off of the NAM subunits are these short chains of amino acids. That's what the ALA and the GLU stand for. Okay, short strands of amino acids. We have to join those strands together with a peptide bond in order to make these cross bridges in the places where there's new cell wall. That transpeptidation step is critical, because if you don't build the new peptide bonds, the new cell wall won't have any strength to it. It turns out that penicillin interferes right here. Penicillin interferes with transpeptidation. So when a human consumes penicillin, the bacterial infection that's going on in their body is sort of stopped, at least in terms of its ability to make any new bacterial cells. The bacteria that exist at the time that the penicillin is administered can no longer divide and create living daughter cells because they cannot finish building the new cell wall on those daughter cells. So with penicillin, the autolysins continue to cut the old cell wall, and the bactoprenol continues to carry new NAG and NAM out to the periplasmic space. And you get the transglycosolases putting the NAG and NAM into the cut places, but you don't get the final step done. And without the final step, those daughter cells simply can't survive. I want to switch gears now and leave the subject of binary fission and start talking about bacterial populations and how bacterial populations grow. Remember, when we have a culture in the laboratory, we have many different individual parent cells in that culture. And as each one of those parent cells starts to go through binary fission, creating daughter cells, we're going to see that whole population of cells increase in size. And again, that's what we refer to as growth when we talk about bacteria. It's an increase in the number of the cells in the population. One cell becomes two, two become four, four become eight. With each doubling, we talk about one generation passing, and the time it takes for that generation to pass is called the generation time. As we watch these parent cells divide in culture, we can see exponential growth occurring over time. So let's take a minute and look at that graphically. Over here on the right-hand side of the slide, we can see what we refer to as a growth curve for a culture of bacteria. And again, it's an exponential type of growth because it has this very classic looking shape to the data line. So the way this was produced, this graph was produced, was we plotted on the x-axis time going by, and on the y-axis the number of cells inside that culture. The cell number is going to double each time the generation time passes. So there's a constant interval of time that passes between each doubling of the cells. When we plot our x and y variables, time and number of cells, we see this curve. So we've drawn in on the curve the number of cells that correspond to each one of the purple-blue data points. We started off with one cell, let's say. We went to two, to 4 to 8 to 16 to 32 to 64 to 128 to 256 to 512 to 1024. Once you hit the steep part of an exponential curve, the numbers go up very rapidly. And this again is referred to as exponential growth. Now, sometimes when we're first working with bacteria in the laboratory, we may not know what the generation time is for that particular species of bacteria, but it's okay because we can still determine generation time just by growing them in the incubator. So if we have a species of bacteria that we don't know how long it takes for a generation to pass, we can calculate that by gathering this kind of data, allowing the culture to grow, and then at some interval of time, taking the culture out and determining how many cells are in it. Take a look at this slide. Now, we still see the same x-axis and the same y-axis. We're still collecting the same data. We have some amount of time going by, and then the number of cells in the culture at each one of those time units. But instead of seeing an exponential shaped curve here, now we have a linear straight line curve. The difference is that we've taken the same data that you saw on this curve, and we've plotted it on a special kind of paper. If you recall from perhaps your high school math classes, this is called logarithmic paper. And when you use it to graph this kind of data, you can take an exponential data set and you can convert it into a linear data set. Same data, just put onto a different type of graph paper, and it turns an exponential curve into essentially the log of that curve, and you get a straight line. Now you might say, well, why would you do that, Dr. Piscopo? That doesn't seem like something I would ordinarily do. Well, if you can also recall from your high school math class, if you have a straight line function on a graph, you can calculate the slope of that line. You can choose any 2 points on the graph, and if you determine the rise or the change in the y value over the run or the change in the x, you can calculate the slope. And it's the slope of that line that will allow us to determine the generation time. Now, there are lots of published tables in textbooks that tell us what the generation time is for the most commonly encountered bacteria that we deal with, as well as other microbes, including things like algae and protozoa and fungi. This table comes out of a textbook like that. You can see that the organism is named over here on the left. The optimal growing temperature is listed, and then the generation time in hours. So, for example, if you look at this E. coli listing, we have a temperature, an optimal temperature of 40 degrees centigrade in the incubator, and we can expect a generation time of about 0.35 hours, or about a third of an hour, or 20 minutes. So that information is helpful to me in the laboratory because I know under what conditions I can grow E. coli effectively. So each species of microbe has its own generation time, essentially. Although in general, we can count on a culture taking about 24 hours to produce colonies for us or a nice lawn of bacteria for us on a petri dish, or to produce enough cells for us in a broth culture that we'll be able to do some work with it. There are some species that require a little bit longer maybe as long as 48 hours in the incubator. And there are some species that you should take out of the incubator sooner, maybe at about 18 hours of incubation. And you learn that from the generation times that are published for each species. Now, something else that you need to know is this. Not only are generation times species dependent, but they're also condition dependent. This is particularly important to us if we're trying to grow them in the lab. Now, what do I mean by condition dependent? Well, for one thing, the temperature will impact the generation time. If E. coli grows best at 40 degrees centigrade and has a generation time of about 20 minutes, if I try to grow E. coli at room temperature, the generation time is going to be longer. It's going to take longer for those cells to divide. So temperature plays a big role in how long the generation time will be. pH is important. The pH of the culture media that we're trying to grow the bacteria in, how much water is available is very important. How many nutrients are available, very important. And also how much waste has accumulated. Remember, inside a bacterial culture, it's sort of a closed system. And over time, all those bacterial cells, they're making waste materials and they're excreting them into the culture container. So all of that will impact how well they're able to divide and how long it takes that division to occur.